Hello, AP Chem students, and welcome to topic 4.6, Introduction to Titration. So the learning objective here is 4.6.a, identify the equivalence point in a titration based on the amounts of the titrant and analyte, assuming the titration reaction goes to completion. So here we go, let's get into it. So we've previously discussed several methods to determine amounts of substances in a solution, including gravimetric analysis. A titration is another laboratory method in which the amount or concentration of a substance can be determined. So this is what this looks like. So we have some very specific glassware here. This is called a burette. There's actually two spellings here. Sometimes you see E-T-T-E, and sometimes you just see E-T at the end. So in your burette goes a known concentration and volume of a substance that you have information about, and we call that the titrant. And then in a conical flask, also called an Erlenmeyer flask, you have the analyte. And this titrand word is not one that we see in AP chemistry. We use analyte. So the analyte is a known volume, but it's an unknown concentration. And so that is what you're analyzing. Now, a burette is well recognized to be very, very precisely measured. And so you can actually get accurate measurements off of a burette to the hundredths place. So you can get 4 significant figures of data here. So in a titration, the species of known concentration, again, is called the analyte. A precisely measured amount of this is added into the flask. I need to move my face up to the top of the screen. There we go. And then the second solution with an unknown concentration is called the titrant. And so then the analyte is carefully and slowly added. And then we usually add the titrant with the burette. Sometimes you see a dropwise titration where you might use like specific amounts like aliquots, and then you would use a pipette at the end. But we'll talk about that a little bit more as we move forward. So the two solutions react in the flask, and more titrant is slowly added until the endpoint has been reached. So this volume is the point in the reaction where it becomes apparent that the equivalence point has been obtained. And so the equivalence point is the point at which all of the analyte has been completely and exactly reacted with the titrant. So you've got that exact amount so that your stoichiometric ratio has been met. So often we used an indicator to make the equivalence point visible. So this word apparent was intentional here. So the indicator is chosen so that it changes color in a range that includes the equivalence point. So this is a chart of indicators, and we'll get much more into indicators later on in the class, but just so that you can see here. So you might, these are pH indicators, and pH titrations is a really common type of titration that we do in AP chemistry. And so these indicators change color based on the pH that of the solution that it's in. So you can see that we have a wide range of indicators here. And so then depending on the species that you're studying, you would choose an indicator that would change color over the pH range at which your equivalence point will be. So now let's continue. So this is what this looks like practically in the lab. So before you reach your endpoint or any point from starting point to well before you reach your endpoint, your solution might be clear. Then you would slow down as you start to see the color linger in solution, but you can still make it go away with swirling. Your endpoint is where you see a very faint color change, and then if you've gone too far, you'd see a vivid color change. So this indicator is called phenolphthalein, which is a really common indicator to use in AP chemistry because it changes color just past the equivalence point. When you see on the graph that I'm about to show you, you'll understand why it's okay that it's just past the equivalence point. So when the reaction has reached the equivalence point, the fact that your moles of titrant is equal to your moles of analyte can be used in conjunction with the formula for molarity to determine the molarity of your unknown. So this looks exactly like your dilution formula. So this is MAVA is equal to MBVB. So in this case, A is acid and B is base, but you could use the same formula even if you were not doing an acid-base titration. So the other type of titration that we see in AP chemistry is called a redox titration. We're going to get into redox reactions and what they are starting in the next section here. So in this equation, MA is your molarity of acid, VA is your volume of acid, MB is your molarity of base, and VB is your volume of base. Now, it doesn't matter if your acid is the titrant or the analyte, and same thing for your base, because you can still use the same formula because we have equivalent number of moles on both sides. And so it doesn't matter where your unknown is as long as your opposite side is known. So again, this is the same equation we use when we're doing a dilution, and in both dilutions and titration equivalent point calculations, the moles on each side of the equation are equal. And so that's why we can do this. Your moles of A is equal to your moles of B. And that's really, really important about this equivalence point. It's what makes it special. So let's look at what this looks like on a graph. So the most common type of titration, again, is an acid-base titration. And when you're performing an acid-base titration, we graph the pH versus the volume of titrant. And that graph is called a titration curve. And it looks like this. So let me move my face again. We'll go down here this time. So you can see that what happens is this equivalence point falls in the middle of this rapid vertical rise in pH. And so that's the reason why we can use phenolphthalein. So even though phenolphthalein doesn't change color until it reaches a pH of around 10, you can see that that 10 is going to be in that vertical part. And so it doesn't matter if our indicator changes color at 7 or just past it, as long as we are in this vertical rise of the curve, because a literal drop will change the pH here. And so reason phenolphthalein is used commonly is, number one, it's pretty safe. Number two, it's pretty stable. And number three, it's a very obvious color change. You're going from clear to pink. So that's really easy to see. So the equivalence point can be found on a titration curve at the place where the curve changes, where the graph changes curvature, and that's called the inflection point. And if you have not yet studied that in math, you will when you get into pre-cal or calculus. So you can see here that your graph has this change in point where it changes from being, it changes between convex and concave. So it changes concavity here. And so that kind of the middle of the S, that's where your inflection point is, and also where equivalence point is. So then we can use the information on this graph to calculate the concentration of your unknowns. So if we're going to look at this graph here, and so we've got milliliters of 0.1 molar NaOH that's being added to 50 milliliters of HCl. And we don't know the concentration of HCl. So then could we figure that out? And the answer is yes, we can. So we got MAVA is equal to MBVB. Again, that equation that we're going to use. So we don't know the molarity of our acid, but we know we have 50 milliliters of it. We know that the molarity of our base is 0.1 molar, and then we can get the volume of base from the equivalence point. So on this graph, there it is. And so that volume is 50 milliliters. So we can plug that into the equation here and do that math. And we can see that in this case, the molarity of acid was equal to the molarity of base at 0.1 molar. So the equivalence point on a titration curve is where it changes direction. And when you're titrating a strong acid with a strong base, that equivalence point will be at pH equal to 7, because the reaction is forming water and a neutral ionic salt. And we're going to get more into that in a couple of topics from now. I'm still in Unit 7. Beg your pardon, Unit 4. The direction of the curve depends on whether the analyte is an acid or a base, and you can see that. So if it's an acid, we would expect our pH to rise as we add base. And then if your analyte is a base, then we would see our pH fall as we add acid. So then that's it about titrations for now. We're going to talk much more about titrations when we get into Unit 8, which is about acids and bases, and also when we get into Unit 9 when we talk about more about redox reactions. But what should I be able to do with this right now? So you should be able to label a titration setup with the terminology of the titration, analyte, titrant, indicator, burette. You should know the vocabulary terms there and be able to identify the known, the unknown, the consumed and excess species at various points during the reaction. You should be able to perform a titration to the endpoint, and you should be able to use MAVA is equal to MBVB calculations at the equivalence point to determine the molarity of the unknown solution. Now, titrations are often used in lab-based questions, so you should be familiar with the steps to perform a titration. So if you don't get the opportunity to do a titration in class right now, there are really great virtual labs for titration online. So all you have to do is look that up. That's it for this topic, and I'll see you soon for the next one.